Okay, so okay, just updating in other platforms regarding the live. So today we will be going to discuss about brief discussion on chapter 5, 12th standard biology, CBSE, principles of inheritance and variation. Okay, so let me share in other platforms as well. We'll wait for 2 minutes and then we will start. Okay. Fine. So the one who already attended these classes regarding this principles of inheritance and variation would be little repeating, but the main motto of these videos are to brush up your past knowledge, so that within 1 hour we will complete entire chapter revision. Okay, and important highlights you need to remember. All these things we will be taken care of. So that is the main motto of this videos. This brief account of brief discussion on this entire chapters. Okay, just a minute. Okay. Okay then. So please share to your friends as well, so that they will also get benefited. So even after this session or during the session. Okay. Fine. So in the 12th standard CBSE, this unit, that is 7th unit, genetics and evolution, right? It's utmost important unit in the entire syllabus. It has, when we talk about academics 12th standard, this has weightage, more weightage than other you know chapters. But here, just put the marks aside, right? But why these chapters are very important? Because these chapters would would lay a foundation to the other chapters. For example, if you see the molecular base of inheritance, this is this lesson, sixth chapter of 12th standard bio CBSE lays foundation to the understand, but better understand the molecular biology, how this molecular biology, at the level of molecular level, how these genes operate, right? So that we can do the application part in biotechnology. Whatever you take, this strategies for enhancement, food production, etc., all technology what we use, right? Thoroughly to to do some to use techniques, for example, biocontrol agents, right? So all these things we need to apply in the sense this genetics chapter is very essential, right? So especially the fifth and sixth lessons are very essential. And evolution, as we already knew that the evolution chapter is equally important in this genetics, this unit, genetics and evolution. Why is that? Like we have to understand how we evolve, at the same time how this life forms has created. So now we are humans and we have got other organisms out there. So how do we see this life? How do we see these differences in life form? How actually, which forces actually created these differences among organisms? So that we can understand how these genes control a cell at the level of cells, at the level of even organisms. So then you will get a overall idea about this entire unit, entire bio. So that's what this genetics and evolution unit in CBSE is utmost important. And though the evolution chapter looks simple and very small chapter which was given, but the concepts are very much confusing, and sometimes you may go wrong when you're dealing the questions in NEET, right? So you should pay utmost uh, you know attention while you're reading and understanding the concepts, right? So today in this unit we will try to good afternoon Abhiniti, good afternoon Kritik Anna, good afternoon Sweta, good afternoon Kadir, good afternoon. Okay, thanks for joining this live. Okay, so let's begin. It's already 6 minutes. I just waited for to join. So let's begin. So today we're gonna discuss this lesson: principles of inheritance and variation. 12th biology CBS. Okay, so it will be useful for NEET preparation as well as academics understanding, right? Board exams also. So this is one type of the videos where I give brief discussion on the entire chapter. There is one more type of videos out there. They are the same chapter will be dealt part by part one, part two, part two. So those videos also will be uploaded soon. So let's begin with the chapter. So when we talk about this evolution, this genetics, right? We talk about this father of genetics, nothing but this. Mendel, we'll talk about Mendel laws in this. At the same time, we start understanding this genetics with the you know the at beginning of studies of this genetics, right? They doesn't know that these the inheritance pattern, right? The inheritance pattern which we observe today is actually you know controlled by some genes that doesn't know at that point of time. So there are patterns over there. There are some factors. They term, especially the mental termed it as a factor, right? So because the molecular biology is not that developed at that point of time, right? So they just gone with the phenotypic expression, and they didn't really understand what exactly controlling, but they do believe that there are some factors actually controls the expression of phenotype. So with that, we begin this principles of inheritance and variation, and then later advancements we got Watson and Crick, right? Frankly, you guys actually paved a path to understand the DNA structure. So in this principles of inheritance and variation, let's break this chapter, you know, lesson or chapter name itself, principles of inheritance and variation. So we are talking about the inheritance, how these, some traits or the characteristics are actually inherited from generation to generation, to the generation to the generation. And what causes variation? So variation among organisms or at the level of cell, at the level of genes also we need to consider. So how these variations will be created? We'll be talking about recombination, meiosis, right? The reproduction, which leads to the recombination of the recombination shuffling of the genes, right? We will talk much about this recombination and how does this recombination would affect, what are the factors does affect the recombination. For example, linkage. So if you break down this entire chapter name, that is principles of inheritance and variation, we will talk about what are the inheritance patterns we observe, and what are the principles are there in inheritance, understanding inheritance. At the same time, what are the principles do we actually understand in the variation, right? So in this, we begin with the Mendel laws of inheritance, and then we talk about inheritance of single gene or one gene, and then we'll talk about inheritance of two genes. Right? That's what we talk about. This inheritance patterns are studied based upon the crosses and based upon the genetic models that Mendel in this experiments that Mendel choose. For example, it's a pea plant. So based upon the traits, characters, if it is a one trait he is looking for, it is and that character is controlled by one gene, then we talk about how this single gene would inherit. And if it is two genes, two traits, how they are inherited. What are those patterns? And then we'll go and talk. We'll be discussing about the sex determination, mutation theory, mutation, and then finally genetic disorders. Good afternoon, Sai. Hope you guys are fine. Okay. Let's move to this Mendel laws. So there was an introduction where people were not aware of this, especially what exactly, uh, uh, how exactly these characteristics are actually transmitted from parent to the offsprings, right? So there are n number of questions that point of time, because they always at that point of time they always have a question that how come one organism is giving the same organism like the characters, traits, etc. So there should be some connection, right? How this baby, for example, in NCERT they gave an example of elephant. Baby elephant always the adult elephant would always give birth to only baby elephant, not other organism. So there is some relation, or there should be some connection with this. There should be a pattern of inheriting certain traits and characteristics. Then, how much identical offspring is with the parent is what lot of questions at that point of time. So that's how the scientists used to think, and that's how they came up with this a branch of biology called genetics, where we can really understand or deal with this type of questions scientifically. So we'll in this subject we deal with the inheritance pattern as well as variation of characteristics. So inheritance of characters from parent to children. At the same time, how much variation is also created, we will be studying in this subject, that is genetics. So let's see the 5.1, that's Mendel's laws of inheritance, where he speak about why he have chosen the model plant, that is pea plant, right? It has got different characteristics, and here before we really dive into this chapter, we need to know different definitions, or we need to understand different terminology like dominant trait, recessive trait, characters, right? Phenotype, genotype, homozygous, heterozygous, right? So these are the terms you need to really have a clear-cut idea. So before you dive into this topic, so you have chosen pea plant, and it has certain traits, or obviously it has got contrasting traits. So based upon the contrasting traits, Mendel choosing this pea plant. For example, if you take stem height, so stem height has got both contrasting tail, that is tall and dwarf. Flower color, one is violet and the other is white. So there is typical contrasting opposite traits. So that we can really study based upon these contrasting characters, we can really understand the pattern of the inheritance. Like that, we have got almost 7 different traits observed in this pea plant. That's what is a favorite, model plants for the Mendel, so that uh, you know, even he conducted several experiments on this plant. Good afternoon, Asha Krishna. Okay, right. So this is one thing we need to cover, that is why we have chosen the pea plant. And if you this see pea plant, there are different characteristics that specific characteristics that this pea plant possesses, right? So, for example, in plants we take pea plant, and in insect models we take Drosophila as one of the experimental model. So we get pure lines, right? So based upon this pure lines, homozygous pure lines, like pure lines in the sense that genotypes would be the same, like pure tall, and pure short. So let's talk about the inheritance of one gene. So when we talk about inheritance of one gene, how this, what are the different patterns that we really see in this inheritance of one gene, right? So apart from the seven traits, there's this pea plant is actually displaying, there is seven different traits of contrasting traits, that it is very easy to study this genetics, right? This Pisum sativa, even it is this pea plant is chosen for the similar studies because it is easy to grow. So it is annual plant. At the same time, pea plant contains both male and female parts as well, like this stigma and stamen, so that we can do this. What do you say? We can control even the pollination. So these are the different aspects. There's this Mendel have chosen this pea plant, right? So we'll continue further. So coming to the single gene inheritance, we will talk about how this single gene inheritance, especially pure lines, are taken as a parents, like pure tall and pure dwarf. That is capital T, small t. When these two pure lines are crossed, right, and then we get the hybrid F1 generation, where always in F1 generation we get an heterozygous dominant trait. It means that it will have capital T as well as small t. It is not homozygous trait, but it could be in heterozygous. So here in this genetics, right, the genotypes are very much important. So in further, we will be talking about pedigree analysis. It will be more clear. So here we need to understand the traveling of, or this movement of this traits. For example, capital T is a diploid condition, right? Diploid, at the same time homologous chromosomes, right? So two copies are there in one. It means from male side and the female side, small t. So we'll take gametes from it, and then we fuse it or we cross them, and then we get the capital T small t, heterozygous dominant in F1 generation every time. And then we self it. So this capital T small t hybrid would be actually self-pollinated to get the F2 generation, and then we study the ratios. When we do this experiment, we get 3:1 ratio. In this 3:1 ratio, what we get is three tall plants at the same time, one dwarf plant. So to get this genotypes, we can put it in a simple Punnett square. So you can take capital T small t, so that if you put the combinations here, what we get is 3:1 ratio of phenotypic ratio. So here I told, right, phenotypic ratio is something which we see outside, the characteristics, how they look. The stem is something which is tall, stem is which is dwarf. So based upon the physical appearance, we say phenotype. And if we cross it, and then we will, if we can, based upon the combinations we get, we can figure it out. Maybe because based upon the back cross or test cross, we can really figure it out what genotype does this plant has. So when we put this Punnett square, right, the hybrid F1 generation, what we got, that is heterozygous dominant with a selfing. So capital T, small t, capital T, small t. So what we get is homozygous tall and homozygous short, and we get heterozygous tall condition. So that's how we get this genotypic ratios, right? So when you see the genotypic ratio, what we get is 1:2:1. So this is how we can study the pattern of inheritance in single gene. So single trait expression, right? So it is the same example we can be using even in the monohybrid cross examples, right? Yeah. So as I told, right, in this can be mathematically also, we can put in perspective that how these this traits can be 3:1, right? But the genotype is 1:2:1. We can get in mathematical expression also, right? But when we talk about this, the when the genotypes are unknown, what we do is whether we do it test cross or back cross to know, right, what exactly the genotype is, right? So during this typical test cross, what we do is a dominant phenotype, right, is crossed with recessive parent, right? So a dominant phenotype would be crossed with the recessive parent. It means when we there are parents over here and the pure lines. When we get the gametes and when we cross, we get the F1 generation. So this F1 generation offsprings, without self pollinating, without selfing, what we do is we will cross this F1 hybrids with one of the parents which are recessive. Then what happens is it reveals the genotypes, right? Actual genotypes of the plant. So this is what we call it as test cross. So with the in NCERT they took simple example of this white and violet flowers, right? Homozygous recessive condition, white flowers, and we have got violet flowers, and then they test crossed. So when they test crossed, it revealed, so the pattern revealed that yeah, the parents might be in heterozygous condition or the parent might be in homozygous condition. If you see the NCERT book, you will be understanding this example very well. So especially to know the genotype of the parent, we can go with this test cross. So in NCERT book, they took the example of what is uh, you know a violet color flowers. So this color flower can be there in homozygous dominant condition or heterozygous dominant condition, but we don't know. So how can we reveal that whether it is a homozygous condition or heterozygous dominant? So for that matter, we do this test cross, then it reveals the pattern. So if it is heterozygous condition, what we get is violet flowers with the even white flowers also will be formed when this heterozygous dominant violet flowers are crossed with homozygous recessive white flowers. But if this violet flower is in dominant condition, right? Homozygous dominant, that is capital W, capital W. So when we cross this homozygous dominant violet flower with a recessive white flowers, we get every possible combination would be heterozygous dominant character violet flowers. So in this way, we can figure it out whether the parent or whether that particular whether that particular plant are holding whether a heterozygous condition or homozygous condition. This is how we can apply the test cross. So that is one thing. And now we'll talk about the laws. When we talk about laws, we have got two different laws that Mendel talks in monohybrid crosses. Right, single gene inheritance displays two things. One is law of dominance and law of segregation. So law of dominance has got yeah, sorry. Okay, I think you guys are able to follow, right? Dominance. So this law of dominance states that the characters are actually controlled by discrete units called factors, because that point of time they don't know about the genes, DNA sequence, etc. So they're simply known as factors. So these factors move from parent to the offspring, and then this factors occurs in pairs as well. So these genes would occur in pairs, and we know that alleles, right? The alternate form of the gene we call it as allele. So tallness and the dwarfness. Capital T and its allele is contrasting trait would be small t. Like that, violet and white flower. Like that, there is a contrasting traits, and the alternate form of this gene would be, either it might be homozygous or heterozygous condition. It would form alleles. So alternate form of the gene. So these factors do occur in pairs like chromosomes. So if you see that this, when this factors comes in pairs, right? So if one of in within this pair, if one is dominant and one is recessive, always this dominant character would actually mask the expression of recessive trait. As simple as that. So in the presence of capital T and small t, always the capital T would be expressed in the monohybrid cross in the law of dominance. So this is what law of dominance stating. So in the law of dominance, what we tell is when there is a recessive trait, alternate form, and a dominant trait, always this dominant trait would mask the expression of recessive trait. So this is the law of dominance, and then law of segregation states that how this movement of alleles would takes place, right? So it means this alleles would not actually bend, like that the both characters would be actually recovered at F2 generation. So when parents pure lines are crossed, we got F1 generation. So if in F1 generation we see only one of the characteristic phenotype of the parents. If we take pure tall and pure dwarf, pure lines, when we cross them, what happens in F1 generation? Always one dominant character only would be expressed, but there is a recessive parent also that is never expressed in F1 generation. But law of segregation states that when these alleles would not blend with each other, but this alleles, for example, the short dwarf plant allele is there, small t, small t. So this allele would come in F2 generation during the process. So this segregation will be takes place, right? So this pairs of the alleles which are occurring in pairs would segregate individually, and even you may see that they will be expressing in F2 generation. So in F2 generation, you can see that even the recessive parent genotype in the offspring as well as the dominant parent genotype in the offspring. So that's how these alleles would separate, right, without blending each other, without mixing. Blending here meaning is mixing. Right, here we'll be talking about even incomplete dominance. When we talk about there are few flowers we see, we will try to see pink flowers where that this they are partially dominant. It means the incomplete dominance is seen here. Right, when you study this incomplete dominance, the white and red flowers are crossed. Right, when you see these parents are of pure red and pure white, that is capital and smaller. When you take gametes, capital smaller, and then we fuse, or when we pollinate, when we you know made among this. So what happens is in this combination we get F1 generation where capital smaller actually should be a dominant trait. It means it should be expressing red color. But obviously in few flowers, what expressed is the pink color. So pink is something from white to red. There is an intermediate color pattern, right? So because this intermediate color came because of the incomplete dominance. So the ratios would remain. When you talk about the ratios, the phenotypic ratio and the genotypic ratio of this incomplete dominance would be the same. That is 1:2, 2:1 would be the ratio of phenotypic expression. At the same time, genotypic expression also would be the same. It means the capital or small r actually in a dominant condition Capital or smaller means it needs to be heterozygous red dominant color, but here it is pink. So what we observe is capital only forms a red color. Capital or smaller heterozygous dominant condition would express as pink, and smaller would be always recessive white colors. So in this, in a law of incomplete dominance, the ratios are important. So both the ratios would be the same, 1:2:1. So here, if you see, the most confusing concept here is that when NCERT people, when they explain, right, they actually explain about the modified allele. So if you talk about this incomplete dominance, so there is some factors actually, not completely expressed. So what makes this? What makes this incomplete dominant? So if you see this allele or trait or gene is little modified. So how this modification takes place? Theoretically, they give three important points in NCERT. That is, when we talk about modified allele, right? When the allele is modified, it can be responsible for the production of three different types of products. Either normal trait it would be produced, for example, either it should be red, or less than the normal, it should be pink. Or the second one is a non-functional enzyme. We are talking about the gene and its expression. So gene products are always proteins and enzymes, right? So protein can be an enzyme, right? All enzymes are proteins, but not all proteins are not enzymes, right? So whatever enzymes are produced by this gene expression, when the alleles or genes which are controlling the expression of these enzymes, which in turn controlling the expression of phenotype, maybe color, for example, if you take this skin tone, right, polygenic inheritance. So if you take the skin tone, one would be very light to the dark, right? So this is how the traits melanin pigmentation takes place. So if you take this modifying allele concept, right, it is responsible for either expression of normal to the less efficient than the normal, or second one is non-functional enzyme can it be produced, or in the third case no enzyme at all. So likewise we get even diseases, genetic disorders, because of the alleles modified due to several reasons. Maybe one of the reasons should be maybe a mutation as well, right? And the next important aspect we talk in this genetics, hello forceps, right? So in this next aspect is we talk about codominance. So the codominance, we in the codominance we take example of this I gene. So I gene is a basic gene which expresses three different phenotypes. Here we talk about three phenotypes of blood groups, ABO blood group. So I gene, this concept is pretty interesting actually. What you can do is, in my class I gave this as an pedigree analysis of blood groups of family. So we did this exercise for present 12th standard batch, and it came very wonderful. So even you can try at home, and you can pick, you can ask your parents or your grandparents and grandparents what are their blood group, and you can find out their genotypes, and you can even figure it out what is the possibility of your children blood groups as well, right? So there would be combinations as well. We can predict at a certain level. So these I genes are responsible for the expression of the antigens, that is Ig, this nothing but which decides the blood group, that is Ia, Ib, right? So here we can take pedigree analysis at next level when we talk about this this incomplete codominant type of expression, right? So in this codominance, we can actually really determine the ABO blood groups in human beings, because this ABO blood groups are controlled by gene I or I gene, or gene I we say, right? So when you take this red blood cell, you will be finding on the plasma membrane, you will be finding the sugar polymers, right, that protrude from the surface of the red blood cell. So if which makes this, this cell, this RBCs will have, if this has an antigen A, then it would be A type of blood group. So there are different combinations you have it in what is it in this blood groups. Hope you guys are following. So codominance is something which we, yeah. So codominance is something which we, so far what we talked about is about the dominance, right? So the F1 generation is resembling either one of the parents, right? One of the parents, and the second one what we talked about is incomplete dominance, where the trait what we get is in between the parents. Like one is red, one is white. We got the pink colored flowers. And here we are talking about the codominance, where the F1 generation resembles both the parents. It means it takes up these two alleles. So, for example, let's consider this AB blood group. Okay, AB blood group is has an married O blood group. So here we need to know the genotypes of each of this. What is there? Each of this blood group. So if you take IG, IA, so this A blood group can be forming in two different combinations. One is heterozygous condition, one is homozygous condition. Homozygous dominant condition and heterozygous dominant condition. It means IA, or IAI. So in O blood group, you will not find any antigen. It means there is no expression of this A or B antigens. So it would be remaining just II. So if I am a AB blood group and the O blood group is my partner, right? Then when there is a marriage between AB and O, what happens is AB has IA, IB, and O has no antigens. It means I, small I, small. Then we can cross this and we can get the combination of F1 generation. So one small I at the same time IA would come into the offspring. So both the alleles are actually coming into the offspring. That's what makes it codominance, when it is different from incomplete dominance and dominant trait expression. So that's what we talk about, the multiple alleles. This is a multiple alleles concept we need to more focus on. So when we talk about the inheritance of two genes, we talk about this, a very famous and a very, very favorite, type of punnets where we get, that is, round yellow seeds are actually, crossed with winkle green parents. It means both are pure lines. One is dominant trait and one is recessive trait. When we cross them and we get different, you know, genotypic and phenotypic ratios. So just like monohybrid cross, even you can cross this dihybrid cross also, but taking genotypes is a little trickier here. So I will be explaining these tricky parts, that examples and Punnett squares and different problems I will be explaining when I'm dealing this chapter part by part. Now let's finish this outline, right? A brief account. So it will have a phenotypic ratio of 1:3:1, 1:3 9:3:3:1 is the phenotypic ratio we get, where 9 of this whole number of this the number of plants we get during this cross. 9 would be round yellow, 3 would be round green, 3 would be wrinkled yellow, and 1 would be homozygous recessive condition, that is wrinkly green. So this is very easy cross that you can do by Punnett square itself. So here in two gene of inheritance, we talk about law of independent assortment. It means For example, the law of independent assortment is like a boyfriend and girlfriend. Like before marriage, law of independent assortment works. In the sense, I'm just giving an analogy. And after marriage, it will be a linked gene expression. It means if we are before marriage, it's like or just like take friendship. Okay, if it is a friendship, like me going to the movies, that would not really hamper someone, my friend going to movie or not coming to the movie or something or the other. So my trait expression or the segregation is not actually depending upon the partner, the allele, or the other pair, actually other trait. So we independently assort. But let's assume that after marriage, what happens? Obviously, the genes are in linked condition, where especially the link gene I explain as, for example, one of the gene expression would be even depend upon the other trait. So they go in pairs, right? So that's what linked genes we'll talk about a little later. But when we talk about law of independent assortment, where they have a degree of freedom that one trait is not controlled by others. So especially when we talk about dihybrid cross, it would work in that way. Right? Yeah. So that's how what we get is so all these different characters would show different ratios when we talk about law of independent assortment. They assort independent of other traits or other characteristics. So according to this law of independent assortment, right, when two pairs of traits are combined in a hybrid, right? So in two pairs, for example, round and round and yellow. So roundness of a seed is something a different trait from the yellow color, right? So these two would actually segregate one pair of characters is independent upon the other pair of the characteristics. So this roundness is nothing to do with the color of the seed. So this round can be green or yellow. This wrinkle seed can be green or yellow again. So that's how this pair of trait, the pair of characters, are nothing to do with other pair of characteristics. That's what independent assortment. And this is restricted to non-linked genes. When the linked genes, when you study linked genes, right, this cannot be applied. So this is limitation of Mendel's law of independent assortment. And then we talk about chromosomal theory of inheritance, like how does this chromosome segregate during meiosis. So when we take a chromosome, within the chromosome, these genes and the alleles will be placed, and they occur both in pairs. Alleles occur in pairs and chromosome occur in pairs, and they segregate individually during meiosis. So that's the thing you need to understand. So the behavior of this chromosome we will be dealing in lesson cell division, right? Especially in meiosis 1, we'll be dealing how these chromosomes would behave during the cell division. So and then comes the linkage concept. Before which we talk about this linkage concept, we will be dealing with the help of one of the genetics favorite organism, that is Drosophila melanogaster, right? So it has certain traits that it has got different wing sizes and body size, color of the eyes. So these are the different traits that this Drosophila we can experimentally get the results, and we can really study different variations as well. So when we talk about the linkage, so linkage and recombinations are well connected. It means when the linkage is more, obviously it is inversely proportional to the recombination, right? So when the linkage is more, the recombination would be less. Why? What? The linkage concept is something which we talk about the presence of this location of the genes, exactly in the loci where exactly they are present. right? The distance, the physical association or physical distance between the two genes. So when they are very close and when they are in link during the meiosis and recombinations, they form very less recombinants. They will be more resembling the parental types. So more higher the linkage, less the distance. So what happens is the recombination would be coming lower. So the formation of recombinants would be less. So that's what inversely proportional. So that for that they gave one example that you can we already discussed in the class itself. So in NCERT, what they did was like they didn't give proper gametes for this cross A cross B, that is 5.11 actually. This cross A cross B, they didn't take the gametes. The gametes they didn't give. If many of the students would get confused directly. Whoever mugs up, right? So whoever by heart this process also, they can write whatever what is there in it, they will print. That's a different story. But whoever really reads and understands, they get stuck at this point juncture where there is no gametes here. So directly they put the F1 generation. So I will in the detailed discussion of this lesson, I'll give the gametes as well for the 5.11 diagram, right, where the cross A cross B are there, which are very important to the understanding of the whole concept linkage, right? And then comes to the sex determination. So in this sex determination, we have different types of sex determination systems, and this is very interesting concept that anyone can teach and anyone can learn. So based upon the sex chromosomes, right? So this the sexes of these organisms are determined. So like that, we will be having two types of sex determination systems, in the sense male heterogamety and female heterogamety. Male heterogamety, it means male is responsible for the formation of two types of gametes. For example, you take humans, XXXY. So in the male, the sperms are of two types. So that's what we say male heterogamety. So when the sperms are actually of two types, right? So these sperms are controlling the fertilization. There's a 50-50 chances that the male or female can form baby, right? So but a lot of illiteracy or lack of awareness causes the blame factor, blames the society blames the female, that especially bearing female child is discouraged in India, and there are stringent rules were taken. There were ban on MTP procedures, etc. But before even that, right, they used to be blaming that the actual cause of the female fetus birth is due to female. But here, if you consider in humans, it is based upon the male, which is determining the sex of the fetus. Especially, there is always a 50-50 chances that male to female ratio. So this heterogamety is observed in the male. It means two types of gametes are formed. One is male, and one has X chromosome, one has Y chromosome. So, but in female, it has got always XX, right? So when this Y chromosome comes and fuses with this X chromosome, then it would be XY, then male. So there is a 50-50 chances. If you ask me, that neither of them shouldn't be blamed off. That should be the level of understanding of these things. It's not that I'm not suggesting that okay, now let's blame the male, or now let's blame the female. No, let's not have that sort of understanding of having a female child, right? We should be taking both or equal. That's what gender sensitivity is. We'll discuss these type of issues in social discussion forum, is one of the type of videos I will be releasing. So let's continue with this determination, sex determination. So if you take these different types of examples in sex determination system, we have got humans. How does this sex determination works? At the same time, we have one of the what is a wonderful examples of sex determination system we see in honeybee, right? So it's based upon not on sex chromosomes, but based upon fertilized or unfertilized eggs, right? So apart from this, in the male heterogamety, for example, you'll be you'll be having different types of male gametes are formed in female heterogamety. You'll be finding different type of gametes are formed in female. It means female gametes will be different. For example, ZW, right? So ZW system would be female heterogamety. So once you can go through the different examples in insects like grasshoppers, cockroach, where do you find? Just do a homework. So let's move on and let's talk about mutation. We will be talking more on mutation on chapter 6. So here they're just explaining the Mendel to make more sense of the Mendel disorders. They just introduce the topic mutation here, but the more mutation is in depth dealt in the chapter 6. Frame shift mutation and point mutation, substitution, addition, and deletion. All these type of mutations will be dealt very clearly in chapter 6, that is molecular basis of inheritance, right? So we'll be talking about mutagens, both chemical as well as physical mutagens, etc. So when we talk about genetic disorders, we come across this pedigree analysis, where we can when I talk about this lesson in depth, I'll be giving some examples also how to deal with this pedigrees. For example, autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. There are different traits or different characteristics that we look when we solving the pedigree charts, right? So we can be a geneticians when we see this pedigree analysis, and when we follow this simple 4-5 rules, we can really analyze any sort of pedigrees. So when we talk about these disorders, we have two types of disorders. One is Mendelian disorders, and then we have chromosomal disorders. So in Mendelian disorders, we will talk about the pedigree analysis at the same time, hemophilia, and then sickle cell anemia. If you take this hemophilia, it is a sex-linked recessive disease. It means the recessive means it is not seen in every generation, and female would be a carrier. So this carrier system, and male is affected, female is affected, male is normal, female is normal, right? In which type of the genetic disorders? For example, there are two types. One is autosomal, and the other one is sex-linked. So in sex-linked, there will be any male carriers. Carrier is a carrier is someone who actually carries the gene which causes the disease, but they don't express that phenotype. But once they get married to a normal or other carrier or other affected person, their offsprings also will get affected. In X-linked inheritance, mostly the females, especially the mothers, carries this genes will not express in them, but they will be expressed in the offsprings, especially the male child. But in the case of autosomal type of a diseases, you can find even male can be a heterozygous carrier, but not in sex-linked type of diseases. So here we talk about the sickle cell anemia, which is a qualitative genetic disorder where there is a single point mutation at this 6th position of hemoglobin peptide. So globin peptide, which results in the changing of amino acids. So that makes this qualitative change, right? When this qualitative change occurs, the normal functions of the RBC would be hampered, and eventually it forms the circular disc biconcave structure of the RBC would eventually turn to be a sickle cell, and it has adverse effects on the health of the person who is suffering with sickle cell anemia. And there are some aspects which actually benefit for them, that malarial parasite cannot really take over these RBCs, and they will be resistant to this malarial parasites infection. So if you see this here in sickle cell anemia, you need to remember the codons, right? Especially the GAG is actually converted to GUG, right? So those things at the 6th position of hemoglobin chain, that you need to remember. So like that, we have got phenylketonuria, which is a metabolic disorder, right? It is an autosomal recessive trait. It means, again, it cannot be seen in every generation. So when we say dominant, it means in every generation this disease is expressed. So likewise, during our in-depth analysis or in-depth detailed discussion on this chapter, I will be taking up the event process and combinations also. Right now, let's move to the further disorders, that is chromosomal disorders. the chromosomal disorders are of it means during the cell division, right? During the cell division, what happens is non-disjunction, or there would be an improper segregation of the chromatids, right? Which results in either gain or loss. So if a chromosome is lost in one cell, it means that it gained in some other cell. So in the either way, both cells are, right, they are abnormal. So this condition we call it as aneuploidy, right? So we have got different examples. We have given, they have given in NCERT that, for example, if you take this uh, Down syndrome, right, we'll be seeing yeah. If you see in Down syndrome, you will be seeing the trisomy. It means the 21st chromosome, autosome, right? It will be having one extra copy of an chromosome. And there is another phenomenon called polyploidy or euploidy, which is often seen in plants, right? And you have other example, that is Klinefelter syndrome. Klinefelter syndrome is what the extra copy of X chromosome, so that XXY. So one extra copy is there, so that the male displays the female characteristics. So ultimately, you will be infertile, and this condition is otherwise called as gynecomastia. And finally, we talk about the Turner syndrome. Turner syndrome, where this is a disorder in which there is a absence of one of the X chromosome in the female. It means XO. So there is no one more X chromosome. So the female may not develop the necessary secondary sexual characteristics, or there will be ovaries would be rudimentary, and obviously the female would be sterile. So these are the different characteristics of the when you talk about the chromosomal disorders. So when we read this lesson, you need to just practice, just like how you practice math. Because when I take this in-depth chapters, right? So that is when I will be explaining with the charts, pedigree. I will take few pedigrees, and I will be explaining those pedigrees as well, right? For example, if you take hemophilia, if father is normal, right, and female is affected. If father is normal, female is normal. If father is normal and female is carrier, what are the combination do we see in offsprings? And we can even find out the F2 generation, F3 generation also. So that is more interesting to few of my students, and they really, we get disappointed when these pedigree questions they were not asked in even CBSE board. One of my student always says that, sir, I'm looking forward to such type of questions like pedigree analysis, but I'm very disappointed that the CBSE board didn't ask. But yeah, maybe neat exam, you guys may find this pedigree analysis, but we should not take any chance, right? Okay, this is during my studies, right, my 10th standard and all, okay, this chapter no need, okay, it will not cover, it is not important, right? That in that way I studied, right? Like, that will not help in long run. Okay, whether it is important or not important, don't read in exam perspective. When it comes to at the beginning level, so when we are preparing for any competitive exam, that is the final battle, right? So you have to have a proper strategy to really meet that criteria. Okay, so at the beginning itself, you should not omit anything. So later parts, you will get an idea that which concepts you need to spend more time, and which concepts you don't need to spend that much of time. This priority should be there, but you should not leave any concept in the NCERT, because NCERT syllabus is very small. The quantity of the syllabus is less, but the quality is good. But still, I expect few more additional content should be added, and few things should be deleted. For example, morphology. When we talk about this plant morphology, right? We talk about this Liliaceae, Asteraceae, Fabaceae, and their floral diagrams. All these things should be removed. Only floral formulas can be given where there is no use for the pre-medical board, whoever writing. These are all degree contents, which only who takes this BSc as botany, right? They, for them only it will be useful, unless or until these topics needs to be removed. That is what I few things to remember. Okay, so that's all for today, and thank you for joining in this live for almost 1 hour, and please share, and before you leave, like this video. At the same time, you share this video for your other biology students, and don't think about whether they are metric or CBSE or any other board. Just share them, share share this video to them, right? Whoever interested in botany, might be a teachers, might be your parents, or might be whoever it is. Let them have this kind of sort of knowledge we can spread, right? So please do share, subscribe to the channel, and share as much as you can. And thank you guys, and see you in the next brief session tomorrow. Thank you guys. Be at home, be safe, take care. So if you have any doubts, you can ask, or I can end the session.